Hello, everyone. Today we're continuing our deep dive of Richard Dawkins and Yan Wong's book, The Ancestor's Tale. In this episode, we're going to discuss how race isn't biologically real and isn't useful in medicine. Now, as you know, I don't pick the order or topics of these tales. They were laid out in the aforementioned book in 2016. However, this particular tale is coming at a rather opportune time because, as I speak, former President Donald Trump and his running mate J.D. Vance are unconscionably spreading utterly false rumors about legal Haitian immigrants in Springfield, Ohio.
In Springfield, they're eating the dogs, the people that came in. They're eating the cats. They're eating the pets of the people that live there.
The American media totally ignored this stuff until Donald Trump and I started talking about cat memes. If I have to, create stories so that the American media actually pays attention to the suffering of the American people, then that's what I'm going to do, Dana.
Vance has claimed that Haitians are eating pets, that they are increasing violent crime, and that they are spreading communicable diseases, all of which have been debunked by the town's law enforcement and mayor. So even though race has no basis in biology, it very clearly still matters to some people. Let's jump right in. Before we get into the tale, I want to announce one recent piece of interesting arthropod paleontology. At the end of July, a new arthropod genus and species was described, UT wanshi, hailing from China and dating from 521 to 509 million years ago. Much like its ancestors we discussed in the brine shrimp's tale, this tiny animal has a series of paired lobopods as well as an onychophoran-like hemolymph system. However, it also has a ventral mouth and lacks dorsal nodes, making it more derived than Carrigmachella. Its lack of sclerotization and segmented appendages makes it less derived than radiodonts and crown arthropods, making it just one more transitional arthropod from the Cambrian. Now, onto the tale. Grasshoppers, crickets, locusts, and katydids comprise the insect order Orthoptera, which means straight wing because of how their wings stick out when folded up. In general, the members of this order have long hind legs with the relevant musculature adapted for jumping long distances. Relevant to today's tale are two closely related species of European grasshopper, Cortypus bruneus and Cortypus bigatulus. In the wild, these two species rarely interbreed because the males of each species attract their respective females with species-specific songs. In captivity, however, researchers coax females of one species to mate with a male of another by playing the song of the female's species. When they do mate, healthy and fertile hybrids are produced. Similar experiments have been done with crickets because crickets have species-specific chirp frequencies. The number of chirps is influenced by temperature, but so is the female's perception of the chirps. In each species, both the number of chirps and the female's perception co-vary, preventing hybridization among species. In experiments, when a female cricket is offered her species' song at different temperatures, she will pick the song that is most similar to one that would occur at her personal temperature. Likewise, Cortypus songs are temperature dependent. The 1987 paper, Separate Localization of Sound Recognizing and Sound Producing Neural Mechanisms in a Grasshopper, describes how a team of researchers attached tiny heaters and thermometers to the grasshopper's head and thorax, being able to heat the head without heating the thorax and vice versa. The researchers found that the temperature of the female's head determines her song preference, while the temperature of the male's thorax determines what song he plays. Of course, both the head and the thorax are generally the same exact temperature in the wild, so hybridization doesn't occur. As it happens, two populations that are considered distinct species in the wild may be induced to hybridize under lab conditions. For example, the beetle species Epilachna niponica prefers to feed on thistle, while the species Epilachna yasutomii feeds on blue cohosh. As a result, in nature, they never interbreed. Under lab conditions, though, the two species can form perfectly fertile hybrids. The same is true for our grasshopper species mentioned earlier. These examples are quite different from hybrids like mules or ligers, where offspring are nearly always sterile. For the grasshoppers, there is seemingly one barrier to reproduction: the song. For a refresher on reproductive barriers, see the narrowmouth's tail. So, how does this apply to human races? For starters, race, unlike species, doesn't really have a definition. The species concepts, though fuzzy, at least provide an objective starting point for a discussion. For the biological species concept, we can talk about interbreeding between populations. In the salamander's tail, we discussed how the endpoints of the Insatina ring species cannot interbreed, while the arms of the ring can. If we apply this concept to humans, then all humans can interbreed and produce fertile offspring, i.e., they are the same species. Even Charles Darwin knew this back in 1871. All the taxonomic ranks above species, genera, family, orders, classes, phylum, etc., lack specific definitions, as do taxa below the species, such as races and varieties. Demes, or local breeding populations, are considered the lowest grouping of some individuals, but demes aren't used as a subdivision in taxonomy. It's better understood as a description of multi-individual interactions, which change constantly. Ecotype is another category used below species, but this can't apply to races either, because ecotypes can refer to environmental heterogeneities on very small scales, where there is no significant genetic differentiation. The fact is, there is no single criterion to define separate races or determine how many races there are. And then what do you do with populations that have so-called mixed racial ancestry? First, let's address that humans do indeed divide each other into categories that we call races based on superficial morphology. An individual may have less than 1/8 African ancestry but still be considered black. This is not for no reason. Historically, the US and Australia employed terms like quadroon to designate individuals who had 1/4 African or Aboriginal ancestry, respectively, and 3/4 European ancestry. Homer Plessy was an American man who only had 1/8 African ancestry, but he was still considered black by Louisiana law, which resulted in the disastrous 1896 Supreme Court case Plessy versus Ferguson that upheld the doctrine of separate but equal accommodations for white and black citizens. Ironically, under the Ohio predominance of blood rule, Plessy would have been defined as white. Further, a number of U.S. states had laws criminalizing miscegenation, or so-called race mixing, until 1967, even though a few states still had such laws on the books until 2000. And there's just something deeply ingrained in our human psychology that forces us to divide continuous series into discrete boxes. For example, when we look at a rainbow, we split it into discrete colors: red, yellow, green, blue, but all these colors form a continuous sequence. Perhaps something similar is happening with regard to races. In support of this notion, the 2021 paper Biological Conceptualization of Race writes, quote, "In particular, even though students may have never learned about race explicitly in formal biology classes, there is a high possibility that they may implicitly construct a biological concept of race by themselves while studying biology." Close quote. But even though race has a historical significance, that doesn't make it a biological reality. As we discussed way back in Eve's tale, a population of humans migrated out of Africa 60,000 to 50,000 years ago, and from that population, all Europeans, Middle Easterners, Asians, Australian Aboriginals, and Native Americans resulted. In other words, a person of European ancestry, or what is traditionally described as a white person, is more closely related to a Native American than to someone of African descent. Furthermore, some African lineages are more closely related to Europeans than they are to other African lineages. The Southern African Khoisan people are considered by some recent analyses to be sister to all other humans, meaning that almost any African person is more closely related to a European or Native American or Southeast Asian or Tasmanian Aboriginal than they are to the Khoisan. It's important to point out that this phylogenetic tree is an oversimplification of the relationship among humans. Really, if we were going to include gene flow among the various lineages, human relationships would look more like this figure. In this light, the concept of races already doesn't make biological sense. In the past, some who would certainly be considered creationists today believed that different races of humans were created separately by God, most notably Louis Agassiz. This was a position called polygenism, which modern creationists want to pretend never existed, especially when trying to use Agassiz's opposition to Darwin in the 19th century as a point for their side, while leaving out how furiously racist Agassiz was in a way Darwin was not. Ironically, creationists who deem all races as stemming from the same original biblical pair, Adam and Eve, would be in agreement with Darwin, who likewise argued that all human races share a common ancestor, though connecting that to ancestral primates in Africa in a way no biblical creationist would allow. Darwin correctly pointed out that naturalists had no idea how many races exist. Quote, but the most weighty of all the arguments against treating the races of man as distinct species is that they graduate into each other, independently in many cases, as far as we can judge, of their having intercrossed. Man has been studied more carefully than any other animal, and yet there is the greatest possible diversity amongst capable judges, whether he should be classed as a single species or race, or as two, Viry, as three, Jacquinot, as 4, Kant, 5, Blumenbach, 6, Buffon, 7, Hunter, 8, Agassiz, 11, Pickering, 15, Bory St. Vincent, 16, Desmoulins, 22, Morton, 60, Crawford, or as 63 according to Burke. This diversity of judgment does not prove that the races ought not to be ranked as species, but it shows that they graduate into each other, and that it is hardly possible to discover clear distinctive characters between them. Close quote. The slightly more recent aforementioned 2021 paper points out, quote, racial categories are not reliable because they are based on subjective judgment based on human traits, while racial classification itself shifts according to different cultures, regions, and individuals. In sum, there are no definitive biological traits that can be used to distinguish continuous biological diversity into folk racial categories, close quote. And the 2013 paper, Biological Races in Humans, argues, quote, adaptive traits such as skin color have frequently been used to define races in humans, but such adaptive traits reflect the underlying environmental factor to which they are adaptive and not overall genetic differentiation, and different adaptive traits define discordant groups. There are no objective criteria for choosing one adaptive trait over another to define race. As a consequence, adaptive traits do not define races in humans, close quote. Furthermore, quote, the dark skins of Africans and Melanesians are adaptive to the high UV found in these areas. Because Africans and Melanesians live on opposite sides of the world, they are more highly genetically differentiated than many other human populations despite their similar skin colors. Europeans, who are geographically intermediate between Africa and Melanesia, are likewise intermediate at the molecular genetic level between Africans and Melanesians, even though Europeans have light skins that are adapted to the low UV environment of Europe. Skin color differences in humans are not a reliable indicator of overall genetic differentiation or evolutionary history, close quote. The paper further comments, quote, even if race is culturally real, that does not mean that these societal racial categories are biologically meaningful. For example, individuals who classify themselves as white in Brazil are often considered black in the USA, close quote. In support of this, the 2009 paper, Color, Race, and Genomic Ancestry in Brazil, found that there was extensive overlap in African ancestry among individuals self-identified as white, brown, or black. Some Brazilian white individuals were found to have more African ancestry than some American black individuals. So if morphology is no help, what about genetics? No again. As the 2004 paper, Genetic Variation, Classification, and Rates, notes, quote, the average proportion of nucleotide differences between a randomly chosen pair of humans, i.e. average nucleotide diversity or pi, is consistently estimated to lie between 1 in 1,000 and 1 in 1,500. This proportion is low compared with those of many other species from fruit flies to chimpanzees, reflecting the recent origin of our species from a small founding population. The pi value for Homo sapiens can be put into perspective by considering that humans differ from chimpanzees at only 1 in 100 nucleotides on average. Because there are approximately 3 billion nucleotide base pairs in the haploid human genome, each pair of humans differs on average by 2 to 3 million base pairs, close quote. The aforementioned 2013 paper notes that historically races have been linked with subspecies, which can be defined as, quote, geographically circumscribed populations within a species that have sharp boundaries that separate them from the remainder of the species, close quote. From there, quote, one commonly used threshold is that two populations with sharp boundaries are considered to be different races if 25% or more of the genetic variability that they collectively share is found as between population differences, close quote. Obviously, this is an arbitrary cutoff, but it serves as a starting point. Applying this criterion to chimpanzees, there are 3 subspecies or races. Pan troglodytes varus in the Upper Guinea region of Western Africa, Pan troglodytes elioti in the Gulf of Guinea region, southern Nigeria and western Cameroon, and all other equatorial chimps. What about applying the same criterion to humans? Applying the same test to humans, the genetic variation between populations is about 4.3%, which is obviously below 25%. By contrast, the vast majority of genetic variation, 93.2%, is found within populations. The same paper concludes, quote, there are no indications of the discontinuities expected when sharp geographical boundaries of genetic differentiation exist. A more detailed analysis reveals that the spatial patterns of human genetic variation are explained well by a series of long-range migrations and population founder events coupled with gene flow with isolation by distance. The gene flow arising from long-range migrations and isolation by distance has obscured any sharp boundaries that may have temporarily existed after the founder events, as well as has reduced the quantitative amount of genetic differentiation. Consequently, neither aspect of the threshold definition is satisfied. There are no sharp boundaries separating human populations, and the degree of genetic differentiation among human groups, even at the continental level, is extremely low. Using the threshold definition, there are no races in humans, close quote. This conclusion is reinforced by the 2007 paper, Genetic Similarities Within and Between Human Populations, that says, quote, the fact that, given enough genetic data, individuals can be correctly assigned to their populations of origin is compatible with the observation that most human genetic variation is found within populations, not between them. It is also compatible with our finding that even when the most distinct populations are considered and hundreds of loci are used, individuals are frequently more similar to members of other populations than to members of their own population, close quote. Even when changing the definition of race to distinct evolutionary lineages within a species, races don't appear. The 2013 paper writes, quote, as pointed out earlier, figure 2 reveals that human genetic distances fit an isolation by distance model. Consequently, it is not surprising that when Behar et al. 2010 sampled Old World populations more finely and used the computer program structure, most individuals showed significant genetic inputs from 2 or more populations, indicating that most human individuals have mixed ancestries and do not belong to a pure group. The races, so apparent to many who cited Rosenberg et al. 2002, simply disappeared with better sampling. These results in Figure 2 further falsify the hypothesis that humans are subdivided into evolutionary lineages, close quote. A bit later it says, quote, the null hypothesis of no admixture between populations is overwhelmingly rejected for this expansion event with a probability level less than 10 to the negative 17th. As noted earlier, the inference of admixture has since been directly supported by studies on fossil DNA of archaic Eurasian populations, close quote. There is some debate over whether race is still a useful term in medicine. However, this is controversial. For example, quote, another adaptive trait in humans is resistance to malaria, which is widespread in African populations. However, malaria is also common in some areas outside of Africa, and malarial resistance is found in many European and Asian populations as well. Indeed, one of the alleles underlying malarial resistance, the sickle cell allele, has its highest frequency in certain populations on the Arabian Peninsula and in India, despite being frequently regarded as a disease of blacks. Close quote. The 2020 paper, We Should Abandon Race as a Biological Category in Biomedical Research, further argues, quote, it is problematic when secondary analyses with respect to race lead to the misinterpretation that biological differences are the true cause for differential health outcomes when only correlations were identified in these studies. We suggest that racial differences in health outcomes are instead frequently a result of multiple testing or reflect socio-demographic causes that are wrongly interpreted as biological. By including race in the assessment of basic data and by considering it in subsequent healthcare choices, race-based medicine is propagated. Without a clear understanding of the causes of racial differences in outcomes in a specific health context, it is not adequate to adapt different treatment recommendations. Doing so can harm patients and exacerbate health inequalities. Close quote. One example the paper cites is a calculator which estimates success rates for vaginal birth among women with a previous cesarean delivery and uses race or ethnicity as a correction factor. Women identified as African American or Hispanic are systematically assigned lower chances of success for vaginal birth following cesarean delivery than white women. However, a number of non-race variables are also correlated with successful vaginal birth following cesarean delivery, including insurance, age, BMI, and whether the woman was married or not. Obviously, these factors should be considered too, and race can't be used as a good proxy for socioeconomic and sociocultural factors. Despite all of this data indicating that races aren't real, or in many cases even practically useful, a, let's say, problematic paper came out in February of this year titled Genomic Data in the All of Us Research Program that, in part, attempted to relate the issues of race, ethnicity, and ancestry. The result was catastrophe. Figure 2 of the paper states that there are 7 categories of race: Asian, Black or African American, Middle Easterner or North African, Native Hawaiian or other Pacific Islander, white, more than one population, and no information. Meanwhile, there are 6 categories for ancestry: African, East Asian, South Asian, West Asian, European, and American, which is apparently supposed to correspond with Hispanic or Latino. Unfortunately, the color scheme between the two legends suggests a 1-to-1 correlation between the races and ancestry, which, as we've already seen, simply doesn't exist. As computational biologist Lior Pachter points out, quote, the authors may have been confused on this point because in the paper they write, of the participants with genomic data in All of Us, 45.92% self-identified as a non-European race or ethnicity. This makes no sense, because none of the race categories are European, and European is also not an ethnicity category. Therefore, non-European does not make sense as either a race or ethnicity category. The authors seem to have assumed that white equals European, as indicated by their color scheme, and therefore non-European race is non-white. By that logic, Hispanic or Latino equals American would mean that Hispanic or Latino is not European, which implies that Hispanic or Latino is not white, contradicting the specific definition of race and ethnicity categories by the Office of Management and Budget. An individual's ethnic self-identification is independent of their race self-identification, and someone may self-identify as white and Hispanic or Latino, close quote. Another problem with the paper is that the authors took essentially continuous genetic data from individuals represented by the panels on the right, split the individuals into discrete ancestry categories, and then used a program called Uniform Manifold Approximation and Projection, or UMAP, to visualize the relationships among those categories. This program is, to put it mildly, inappropriate for the job because the point of UMAP is to, quoting friend of the channel Zach Hancock here, exaggerate every difference to make them look as discrete as possible for ease of visualization. When population geneticist Graham Coop plotted the same data points but with principal component analysis, a very different looking chart appeared. The reason for this, Coop explains, is, quote, in particular, many of the people classified as of African ancestry have recent European ancestry, and Latino/admixed refers to a mix of indigenous American, West African, and European genetic ancestries. Yet, European ancestry is held apart as a separate group. Close quote. Before we close today, let's kick one more hornet's nest because, why not? Is there an intelligence gap between races? Well, if races aren't a biological reality, as we've seen, then of course no. Or at the very least, any differences observed are not due to biological factors. They are a consequence of different socioeconomic conditions. But let's dig deeper. Relating to the discussion of IQ and races is what's called the hereditarian hypothesis, which proposes two claims. A, genetic differences between races substantially contribute to differences in cognitive ability as measured by IQ tests, which are impervious to alterations in the environment. And B, that the relevant genetic differences between races were produced by divergent natural selection that favored higher cognitive ability outside of Africa. In 2021, evolutionary biologist Kevin Bird used genomic data from the 1000 Genomes Project to test both hypotheses. First, Bird found that alleles associated with high educational attainment and cognitive performance do not show more genetic differentiation than control single nucleotide polymorphisms not associated with either. And Bird could detect no evidence of positive selection for these traits. So now we come to the end of our tale. There is no biological evidence for human races, they are not useful in medicine, and there is no link between race and IQ. I want to close with the words of geneticist Alan Templeton. Quote, "Humans are an amazingly diverse species, but this diversity is not due to a finite number of subtypes or races. Rather, the vast majority of human genetic diversity reflects local adaptations and, most of all, our individual uniqueness." Close quote. So, thanks for watching, and we'll see you all next time.